Using quite a crude model, I'm going to describe for you now how a tRNA is reactivated and can be reused during translation. So first of all, here we have a tRNA molecule, and as you can see, it no longer has an amino acid attached, so it's dropped it off during the translation process. Here we have the amino acid that is associated with this particular tRNA anticodon, and here we have the specific tRNA activating enzyme that is going to do the job of reattaching the amino acid to the tRNA. So first of all, the amino acid binds to the active site of the tRNA activating enzyme. And as you can see, the active site of the tRNA activating enzyme is complementary to the shape of the amino acid. Then the appropriate tRNA molecule comes along and also binds with the active site in the right orientation for this process to happen, just like with all other enzymes. And then this enzyme is very special. It can break down ATP, so cellular energy. And by breaking down the ATP, it creates a very high energy bond, which in this case is represented by tape. And this high energy bond between the tRNA and the amino acid is created. So this tRNA has now been activated. It has a new amino acid attached, and it can go off and be used in translation again. Now there's one more important concept to understand here, and that is about this high energy bond that's been created between the tRNA and the amino acid. The energy that has been put into this bond is used to form the peptide bond when the amino acid detaches. So when this amino acid detaches from its tRNA and the tRNA goes to get recharged again, this amino acid will use this high energy bond to join with the next amino acid in the sequence during translation. Now I'm just going to show you this again one more time, but with a different amino acid so that you can see how the enzymes are specific. So in this case, exactly the same thing will happen again, but this time we've got a different amino acid. Notice that the tRNA activating enzyme is complementary to the amino acid. The previous tRNA activating enzyme was complementary to that amino acid. This one is complementary to a different amino acid, but it's the same process again. So the amino acid binds to the active site of the enzyme. The tRNA also joins, and then the tRNA activating enzyme creates a high energy bond between the amino acid and the tRNA, and that ineffectually is now charged. Okay, so then in this case, if we were making a sequence, if we were in the P site of the ribosome, this amino acid would detach from the tRNA and attach to the other amino acid in the sequence. So this is how the high energy bond is used. So there are two key things that you need to understand about this process. The first is that the tRNA activating enzymes are complementary to the amino acids, not to the tRNA, and that's because there's more than one tRNA that can code for a given amino acid. So one amino acid might have multiple tRNAs, and so each amino acid has its own enzyme, not each tRNA. And then second of all, the second most important thing to understand about this process is that the tRNA activating enzyme creates a high energy bond. So you need an input of energy to make the bond between the amino acid and the tRNA, and that is so that the energy can be used to form the peptide bond when it's undergoing translation.